Now we will discuss about the laboratory techniques in the biochemistry. One is flow cytometry. Flow cytometry, its meaning is flow of the cells. Cyto, its meaning is cells. So flow cytometry is there is flow of cells, and it is a lab technique where flow of cell take place and we assess size, granularity and protein expression of individual cells. Definition of flow cytometry: laboratory technique to assess size, granularity and protein expression of individual cells. It is also known as immunophenotype. So its process starts with cells tagged with antibodies specific to surface or intracellular proteins. First, cells are tagged with antibodies and proteins. Antibodies linked to fluorescent dyes. So some antibodies are linked to the fluorescent dyes, and a laser focuses on each cell. In the flow cytometry, laser focuses on each cell. It measures light scatter and fluorescence intensity. Data representation. There are two types of data representations. Data representation. There are two types of data representation: histogram and scatter plot. Histogram shows one measurement, and scatter plot, it shows two measurements. For example, CD3 versus CD8. There are some quadrants, and for example, left lower quadrant negative, it is negative for both CD3 and CD8. Right lower quadrant, it is positive for CD8 and negative for CD3. Left upper quadrant, it is positive for CD3 and negative for CD8. Right upper quadrant, it is positive for both CD3 and CD8. Clinical uses of flow cytometry. Hematologic abnormalities, leukemia and paroxysmal nocturnal hemoglobinuria or PNH, immunodeficiencies, CD4 positive cell count in HIV. So these are clinical uses of flow cytometry. So it is detecting fetal RBCs. It also used for detecting fetal RBCs in the maternal blood. So we have discussed about the flow cytometry. So I hope you can understand it, and comment down below your question. Second technique is microarrays. Definition: microarrays technique to compare gene transcription between two RNA samples. Its application: detect single nucleotide polymorphism or SNPs, detect copy number variants, CNVs. So it is used, microarrays, these are used in genotyping, clinical genetic testing, forensic analysis, cancer mutation studies, genetic linkage analysis. So microarrays are used in the genotyping, clinical genetic testing, forensic analysis, cancer mutation studies, and genetic linkage analysis. ELISA or Enzyme-Linked Immunosorbent Assay. So definition of ELISA is: it is immunologic test that is used to detect specific antigens or antibodies in the blood. Principle: antibody linked to the enzyme, substrate react with the enzyme and produces detectable signals, color fluorescence. And types of ELISA are, and there are two types of ELISA. One is direct ELISA and second is indirect ELISA. Direct ELISA, it uses enzyme labeled primary antibody and it detects antigen directly. And indirect ELISA, what it does, it uses labeled secondary antibody and it detects antibody bound to the antigen. And now we will discuss about the clinical uses of ELISA. So ELISA is basically used for screening for HIV infection. And there are some advantages of ELISA test. So advantages, it is highly sensitive. Its advantages are high sensitivity and specificity. These are two advantages of ELISA. Its limitation, it is less specific than the Western blot. So now we will discuss about the key takeaways and medical terms explained. Flow cytometry. What is flow cytometry? Flow cytometry technique to analyze physical and chemical properties of cells using lasers and fluorescence. Fluorescence is basically a dye. So it is technique to analyze physical and chemical properties of the cells by using lasers and dyes. Immunophenotype. Pattern of proteins expressed on cell's surface that is used to classify cell types. CD3 and CD8. CD3 and CD8, these are cell surface markers. CD3 is found on all T cells. CD8 found on cytotoxic T cells. And leukemia, cancer of blood forming tissues. Leukemia is cancer of blood forming tissues. Paroxysmal nocturnal hemoglobinuria or PNH. It is a rare blood disorder causing RBC's breakdown. So in this disorder, like in paroxysmal nocturnal hemoglobinuria or PNH, red blood cells breakdown takes place at the night. Microarrays. It is tool to study gene expression and genetic variations. So microarrays is basically used for genes testing, to study genes expression and variations in the genes. SNP or single nucleotide polymorphism, variation in a single DNA base. CNV or copy number variant, variation in the number of copies of a DNA segment. ELISA. What is ELISA? It is test to detect antigens or antibodies using enzyme-linked reactions. Western blot. Western blot is more specific test for protein detection that is often used to confirm ELISA results. Now we will discuss about summary of this. Flow cytometry analyzes cell properties and protein markers using lasers and fluorescence. That is crucial in hematology and immunology. Microarrays compare gene expression and detect genetic variations, widely used in research, diagnostics, and forensics. And ELISA detects antigens or antibodies in the blood. It is highly sensitive, that is commonly used for HIV screening, but less specific than the Western blot. So Western blot is more specific than the ELISA.